Hi, we will continue talking about protein digestion, starting from the small intestine. Okay. I'll share my screen with you. Okay, need some adjustment here. I think you can see the screen with a white background here, right? Okay. Okay, protein digestion. Now we are moving to the small intestine. Starting with um, duodenum. Okay. Earlier I mentioned that duodenum is the beginning part of the small intestine, and the pancreatic secretions feed into the lumen of duodenum, and this is the location that just little absorption occurs, and some digestion is being studied. Okay. There's no significant nutrient absorptions really happening here, only except for those the basic units of nutrients such as free amino acid and glucose and so on. Now moving to jejunum, and the jejunum is the main major site for the digestion and absorption. And the ileum is not the main, it's not important for the nutrient digestion absorption in the case of mammals. But in the case of avian species, I already mentioned that we define the ileum for avian is from the macros diverticulum to the ceca. So ileum is a lot longer in the case of avian compared to the mammals. So in the case of avians, both jejunum and ileum are the important site for the nutrient digestion and absorption. Okay? So there's a different the way that we see the structure of the small intestine between the mammals and avian species. Okay, now the digesta are moved from the stomach to the small intestine. It is moved as a bolus. It's not continuous, but a small portion of digesta will be moving going through the pyloric valve and move to the lumen of the duodenum. Okay? And they are very acidic because in the stomach, hydrochloric acid is added, and the pH is maintained around 2 to 4. So it's acid. So we call this digesta as acid chyme. Okay, so acid chyme is passed from the stomach and to the lumen of the duodenum, and that will stimulate intestinal endocrine cells lining the duodenum, and they will cause the secretion of the hormone called secretin or secretin. Okay, so this is the second hormone that we are talking about here. Earlier, we talked about gastrin, and then now we're talking about secretin. The function of this hormone, secretin, is stimulating the pancreatic cells to release hydrogen carbonate or bicarbonate, the same thing, hydrogen carbonate or bicarbonates. So it will start releasing the pancreatic cells will release the bicarbonates. And this bicarbonates will neutralize the acid chyme came from the stomach, so the digesta in the lumen of the duodenum will be neutralized thanks to the secretin here. And the pancreatic cells will also secrete the enzymes, digestive enzymes, to initiate the protein digestion. And those pancreatic secretions in relation to protein digestion, they include both groups of proteases, including endopeptidases and the exopeptidases. We already talked about this too. The endopeptidases include trypsinogen, chymotrypsinogen, proelastase. All three of them, they are inactive form of proteases. So trypsinogen is an inactive form of trypsin. Chymotrypsinogen is an inactive form of chymotrypsin. And the proelastase is an inactive form of elastase. So I already mentioned that we call those inactive enzyme as zymogens. These zymogens need to be activated. Okay? And all these three enzymes, proteases, they belong to serine protease, and which we will talk about in the next few slides later. And there are two exopeptidases will be secreted from the pancreas. One is the procarboxypeptidase type A and type B. Both of them are inactive yet, so they are zymogens. And procarboxypeptidase A will be activated to carboxypeptidase A, and procarboxypeptidase B will be activated to carboxypeptidase B. Okay? So these are the zymogens secreted from the pancreas thanks to the function of secretin. Okay, let's talk about the trypsinogen. So we'll spend some time to talk about each enzymes. Trypsinogen should be activated by this another enzyme called entropeptidase. Entropeptidase. It's not endo or exo, but it's entro. Entro means gastric or intestinal. The entropeptidase. This is also called enterokinase. So that's we are two different terms referring the same enzyme. Okay, this enzyme this enzyme is secreted from the duodenum by another hormone called cholecystokinin, CCK, or cholecystokinin. This is the hormone will cause the release of entropeptidase from the duodenum. Okay? And this enzyme will cleave some portion of amino acid from the amino end of trypsinogen, okay? And that will activate trypsinogen to trypsin. Actually, there's the enterokinase is removing 6 amino acid from the amino end of the trypsinogen, and this trypsinogen will become active to trypsin. Okay, so now trypsin is activated. Okay, so what is the function of trypsin? Moving to next slide. Okay, trypsin as a function is very important protease. Okay, it will cleave peptide bonds at the carboxyl ends of basic amino acids in the middle of protein. Okay, here we have to remember a few things. Okay, amino acid will have a carboxyl end and amino end. The carboxyl end is on your right side, and the amino end is on your left side. That's the agreement we have, okay, in biology. Okay, and then also we need to remember basic amino acid. What are the basic amino acids? Okay, those are the amino acid with additional nitrogen on their R group. So those are arginine, lysine, and histidine. You may include ornithine, but ornithine would not be a part of protein, so I exclude it here. Okay, so we have to check arginine, lysine, and histidine. Thanks to this additional amino group on their R group, they are positively charged, so they are basic amino acid. Okay? So I'm showing you a section, a small portion of a long polypeptide here, and I found the lysine. There's a glycine here. This is just the example. So trypsin will come or look for the R group of the basic amino acid. Okay, in this case, it's lysine here. So this trypsin will look for this amino group on the R group of an amino acid. Okay, so it identifies lysine, and then trypsin will cut the peptide bond to cleave the peptide bond on the carboxyl end of lysine, or carboxyl end of basic amino acid. So this can be lysine or arginine or histidine. So this peptide bond will be cleaved. Okay? That's the function of trypsin. Okay? So if there's 20 basic amino acid in the middle of a polypeptides, trypsin will cut those trypsin peptide bonds on the carboxyl end of those 20 basic amino acid. Right? I hope that makes sense. Right? Now we are moving to another next zymogen called chymotrypsinogen. It sounds similar to trypsinogen, but it's called chymotrypsinogen. Okay, this enzyme zymogen need to be activated to chymotrypsin, but this activation is done by trypsin. Okay, so trypsin is very important enzyme that need to be activated first. How? By cholecystokinin, causing the release of enterokinase, and enterokinase will convert trypsinogen to trypsin, and this trypsin will do its function, but also another important function is the activation of chymotrypsinogen to chymotrypsin. So trypsin need to be activated to activate other proteases. Okay, so now we have a chymotrypsin. Okay, what does this do? This chymotrypsin, once it's activated, it will cleave the peptide bond on the carboxyl side of aromatic amino acids in the middle of protein. Okay, so now we need to know what aromatic amino acids are that we know. What are those? Okay, tryptophan, phenylalanine, tyrosine. Right? So those are aromatic amino acids. Right? So it will look for those aromatic amino acids in the middle of the polypeptide and cut the peptide bonds on the carboxyl side of those amino acids. So that's the function of chymotrypsin. Right? The next is proelastase. This proelastase is also zymogen. It's not active, but need to be activated by trypsin to elastase. And the function of elastase, okay, well, even though it's not fairly non-specific, it prefers to cleave peptide bonds on the carboxyl side of amino acid with small side chains, such as aliphatic amino acids. So what are the aliphatic amino acids? Those are glycine, alanine, valine, isoleucine, and leucine. Okay? Those 5 amino acids, we call them aliphatic amino acid. So as an example, this is a part of a small section of polypeptides. Left is amino end, right is carboxyl end, and elastase will look for the R group of aliphatic amino acid. So here the elastase found the valine, okay? Valine, and valine has 3 carbons as an R group, okay? And it will cleave the peptide bond on the carboxyl end of a valine here. So that's where the elastase will cleave the peptide bond here. Not the other one, but on the carboxyl side, not the amino side, but the carboxyl side. Right? So that's how these 3 endopeptidases work differently: trypsin, chymotrypsin, and elastase. So we talked about 3. So I made a summary here. Trypsin cleaves the peptide bond on carboxyl side of basic amino acid. Chymotrypsin cleaves peptide bond on carboxyl side of aromatic amino acid, and elastase cleaves peptide bonds on carboxyl N, carboxyl side of amino acid with small side chain, including aliphatic amino acid, in the middle of protein. So it's endopeptidases. So if I give you an example of just randomly made polypeptide here, I indicate, I code each peptide bonds by number 1 through 21. So if you look at this one, glutamate has a 2 peptide bond. One is on amino side, another one is a carboxy side, 9 or 10. So now I ask you, okay, this trypsin is coming. Trypsin came to this polypeptide and is working to digest this polypeptide. Okay, which peptide bonds will be cleaved by trypsin, right? So what should you do? You should look for the location of basic amino acid, arginine, lysine, histidine. So, okay, here's arginine. Okay, arginine. What would this trypsin do? Trypsin will look for peptide bond on carboxyl side of arginine. So number two will be cleaved. Another one. Okay, here's a lysine. So which one, number 7 or number 8? Trypsin will cleave number 8, not number 7, but number 8. So this is a lysine peptide bond on the carboxyl side of lysine. So this will be cleaving number 8. And also there's a arginine. Which one, number 10 or 11? Number 11 will be cleaved. And here's another lysine, 14 or 15. The trypsin will cleave number 15 peptide bond, which is on the carboxyl side of lysine and so on. So that's how you can identify where these enzymes will cleave, digest the protein here, right? I can ask question about which peptide bonds will be cleaved by elastase. Oh, you have to look for valine, so number 4. Look for leucine, so number 7, not number 6, but number 7. Valine, number 9, not number 8, but number 9, and so on. Okay, I'm sure you can do this. Now, briefly, it's not the key interest, but briefly, I'd like to introduce different types of proteases. There are 6. Okay, the first group is the proteases using activated water, right? In the hydrolysis. Hydrolysis means utilizing water in the reaction, okay? So these are aspartate protease, glutamate protease, metalloprotease, proteases. Those are 3 groups of proteases, enzymes for protein digestion, using the activated water for the action. Okay? I think that's just enough to know about in this class. We will not learning about physical chemistry here, so this is just enough. But the other 3 types, the proteases that we more commonly see is the cysteine proteases, serine proteases, and threonine proteases, and they use amino acid residues as a neutrophile to attract and attack the peptide bond. So as an example here, I'm showing you a chymotrypsin here. This is just a rough drawing, okay? And chymotrypsin belongs to serine protease, okay? So chymotrypsin is a serine protease. Okay, if you look at this enzyme protein, chymotrypsin is also protein, and there is an active site. This is the active site that is where the enzyme action will happen. Okay, and the active site is made of will have 3 amino acids exposed. Those are aspartate, histidine, and serine. So one of these three amino acids are serine, is a serine. And serine is providing the active site of this enzyme. So we call that serine protease. Okay? That's how the proteases are classified. Okay? I think that's enough to know for our class. So I just wanted to show a little more details about it. But chymotrypsin is a serine protease because you can find the serine in the active site called the catalytic triad. One of the amino acid in triad is serine. Okay? Let's move on. That's enough for this. Okay? I just talked about this serine protease. Chymotrypsin is the example, and trypsin and elastase. So all three endopeptidase secreted from the pancreas is serine proteases. Okay? In the case of pepsin secreted from the chief cell. Okay, it is a aspartic protease using active water as a neutrophile. Right? So it's a very different types of protease, pepsin versus the trios, trypsin, chymotrypsin, and elastase. Okay, let's talk a little about the reasons for the specificity of enzymes and how trypsin know which peptide bond to cleave. Okay? So if you look at the structure of protein, this enzyme trypsin, and there is the substrate binding pocket. There is a specific area very close to the active site. There is a binding pocket. Okay? This binding pocket here, this part, the aspartate is exposed. Aspartate is exposed inside of the this binding pocket. The aspartic acid, the side chain is an extra carboxyl group, is a anion is a negatively charged. So this enzyme will look for basic amino acid with positive charge on their R group. So which means lysine will have an additional amino group on their R group. So it's positively charged. Arginine, same thing. So thanks to aspartate on the binding pocket of trypsin, it will look for the lysine, arginine, or histidine, and then the serine will cause the cutting. Serine and histidine together will cause the cutting of the peptide bond. Okay, so that's how trypsin can be smart to find basic amino acid. In the case of chymotrypsin, okay, its binding pocket is strongly hydrophobic. Very strongly hydrophobic means they are covered by hydrophobic amino acids. So that means amino acid with the hydrophilic R group cannot come. So they have a strong preference to bind to hydrophobic amino acid, which are aromatic amino acid. They are very strong hydrophobic. So those are phenylalanine, tyrosine, and tryptophan. Okay? And then the serine will cause the cutting of the peptide bond on the carboxyl side of these amino acids. Okay? That's how this chymotrypsin can be very specific. In the case of elastase, it's not very specific, but the feature of the binding pocket of elastase is very small, very shallow, right? So they will allow a very small amino acid, the amino acid with a small R group. So those are like alanine, glycine, valine, isoleucine, leucine. Okay, those are commonly called aliphatic amino acid. Okay, that's the reason while elastase will prefer cutting the peptide bond on the carboxyl side of these aliphatic amino acids. Now let's move to exopeptidases. There are two zymogens secreted from the pancreas. Those are procarboxypeptidase A and B. And these zymogens, again, also, it should be activated by trypsin. So trypsin will activate procarboxypeptidase A to carboxypeptidase A. The function of carboxypeptidase A is cleaving peptide bond of the peptide bond at the amino end, no, carboxyl end of aromatic amino acid and aliphatic amino acid. So that means, when chymotrypsin react and digest long polypeptide into smaller peptides, those peptides always start with aromatic amino acids, like tryptophan, tyrosine, and phenylalanine, because chymotrypsin will cut the peptide bond on the carboxyl side of aromatic amino acid. So chymotrypsin will cause the small peptide starting with these aromatic amino acids on their carboxyl end or C-terminal here. Whereas the elastase will cause the production of smaller peptides starting with aliphatic amino acid on their carboxyl end or C-terminal. Okay? And the carboxypeptidase A will remove aromatic amino acids or aliphatic amino acid, which located at the carboxyl end of the small peptide, and this aromatic amino acid and aliphatic amino acid will become free single amino acids. Whereas the carboxypeptidase B, it will look for the basic amino acid on the carboxyl end of smaller peptide. Okay? So if you remember trypsin, this trypsin will cut the peptide bond on the carboxyl side of basic amino acid, lysine, arginine, histidine. So creating the small peptides, starting with arginine, lysine, and histidine. And this carboxypeptidase B will come and remove those lysine, arginine, and histidine from these small peptides. Okay? So those are the functions of carboxypeptidase type A, type B, working together with chymotrypsin, elastase, in the case of carboxypeptidase A, or working with trypsin in the case of carboxypeptidase B. Right? And it is known that proline, the amino acid proline, caused inhibition of this carboxypeptidase A and B. So here, one example here, right? Let's say this green amino acid is lysine, right? So the reason why we have this small peptide starting with lysine on the carboxyl end is because of the outcome of trypsin action. Trypsin cut peptide bonds on the carboxyl side of lysine. So this small peptide is created. And then the carboxypeptidase B will come and cut peptide bond on the amino side of basic amino acid. Okay? So causing the release of this lysine from this peptide. So lysine is now free. It's a single amino acid now. And the single amino acid is small enough and it can be absorbed into through the enterocyte. Yes, that's now the protein digestion is more complete here. So by the action of these five enzymes we talked about, but including pepsin is six. Okay, pepsin in the stomach, and trypsin, chymotrypsin, and elastase, carboxypeptidase type A, type B. All the six enzymes, proteases, will work together and digest a long polypeptide into very small peptides or amino acids. And so the end products from the pancreatic secretions will be amino acid and small peptides. Okay, but small peptides can still be further digested because there are additional enzymes secreted, not from the pancreas, but from the intestinal mucosa. The enterocyte will secrete the enzymes called aminopeptidase or dipeptidase. Aminopeptidase is an exocleaving enzyme, exopeptidase, removing amino acid from the amino end of a small peptide, right? Whereas the dipeptidase is cleaving a peptide bond of dipeptide. Dipeptide is peptide with two amino acids. Right? So if you look at the final stage of digestion and the absorption of the end product, if you see this illustration on your left side, that's the lumen of the small intestine, lumen of the jejunum. The right side is the inside of body. Right? In the lumen, as an outcome of protein digestion, we can see a single amino acid, free amino acids. They'll be absorbed into going through the membrane, cell membrane, and absorb into the enterocyte and entering the blood circulation. And this is done by amino acid transporters called systems. Okay? And dipeptides and tripeptides, okay? And number one, dipeptide with two amino acid, tripeptide with three amino acid. They will be also effectively absorbed into the enterocyte by the transporter called peptide transporters, pept, okay? And in the enterocyte, they can be further digested into single amino acid and entering the blood circulation. The case of tetrapeptide, or a little longer than tetrapeptides, and it will be further digested by aminopeptidase on the brush border of the enterocyte, and it becomes smaller peptide, dipeptide, tripeptide, or single amino acid and being absorbed. Okay? So at the end of protein digestion, it should be single amino acids or dipeptides or tripeptide to be absorbed. Okay? So this is the overview of protein digestion. Acid comes from the stomach and stay in the lumen of the duodenum. They will cause the secretion of cholecystokinin and also secretin from the intestinal endocrine cell found in the duodenum. Okay? And the secretin will cause the secretion of pancreatic juice, including bicarbonates and the zymogens. And cholecystokinin will also cause the secretion of pancreatic contents, including bicarbonates and zymogens. In that way, the function of secretin and cholecystokinin is same. But in addition to that, cholecystokinin will cause the secretion of entropeptidase or enterokinase from intestinal mucosal epithelial cell. And this enterokinase or enteropeptidase will activate trypsinogen to trypsin. But when trypsin is activated, trypsin will cause the activation of the other zymogens. Okay? And now you know the function of these enzymes we just learned. Okay? There are some factors causing the issues with protein digestion. So we'll talk about some of them a little bit here. Okay. Some food ingredient or feed stuff include protein blocking the function of protease enzymes. We call it protease inhibitors. Okay. The famous one, the notorious one, is a trypsin inhibitor. Trypsin inhibitor, very well found in the raw soybeans or legume seed. Okay, there are two different types identified: Kunitz trypsin inhibitor and Baumannburg trypsin inhibitor. Okay, both of them have the same function, blocking the function of trypsin. So they are trypsin inhibitor, but this type of Kunitz trypsin inhibitor, Kunitz TI, that's the majority, the predominant inhibitor. However, it is very weak against heating. So the cooking of soybeans will denature, destroy Kunitz trypsin inhibitor. Whereas Baumannburg trypsin inhibitor, that's minor, but it's heat stable, so the cooking will not destroy Baumannburg trypsin inhibitor, and it will block the function of trypsin. Also, it can block the function of chymotrypsin too. Okay? Some feed ingredients or foodstuff will contain tannins. In the case of sorghum or milo contains tannin. Some vegetables will contain tannins. Okay? And the tannins is polyphenolic compound. So in some way, it can be a very important bioactive beneficial compound. If it is handled correctly, the basic unit of tannin is gallic acid. The gallic acid has a very good anti-oxidative capacity function. Okay? But tannin is a large molecule. What it does is it will combine or bind the protein and make a very big complex structure, and that protein cannot be digested by proteases. So tannin will block the protein, the excess of proteases, and thus it will reduce the protein digestibility. Okay? There's another one called phytate or phytic acid. Okay, that's commonly found in the grains. This phytic acid is based on inositol. Okay, inositol is made of 6 carbons, making a ring, and phosphoric acid are attached to each carbon. So this phytic acid will have a 6 phosphoric acid. Thing is, this bond between phosphorus and the carbon through oxygen, this bond cannot be digested by the endogenous enzyme that we secrete. So those phosphate attached to inositol would not be available for the use by the animal. Right? So phytate is causing the poor use of phosphorus by the animal. But in addition to that, this phosphoric acid attached to the inositol as a part of phytic acid, they'll bind with other minerals, including and as well as the amino acids, right? Especially the amino group on the R group of amino acid, like a lysine, and that can be attached to the phosphoric acid that is negatively charged, and it will cause the will block the action of, like, trypsin cannot find the lysine as an example. Right? So that will reduce the protein digestion, and we have research papers showing that, proving that. Okay? That's showing that phytic acid are causing the reduced protein digestibility. Some other factors are fiber. Here, fiber is very broad a term, but here I'll say non-starch polysaccharide, called in short NSP, NSP, broadly speaking, is similar to fiber, right? But carbohydrate, excluding starch, and non-starch polysaccharide. Okay, among those, some of them are can absorb the water. So it is not entirely correct, but we call it soluble NSP because some types of NSP can absorb water, becomes highly viscous. So then the examples are galactomannans, arabinoxylans, and beta-glucans. They are high in the cereal grains. Okay, so if you use cereal grains and in the food or feed, um, when water is added in the GI tract, that will be highly viscous. Right? So the soluble NSP, when they absorb the water, becomes highly viscous, causing the viscosity of digesta. And when they are viscous, they will entrap. They'll put all other food particles, feed particles, including fat, protein, starch, and minerals, into highly viscous matter. Okay, and then our endogenous enzymes, either lipase or amylase or protease, including trypsin, cannot access to its substrate because they are covered by soluble NSP. So that will reduce the protein digestibility and but also lipid digestibility and um, carbohydrate digestion too. So soluble NSP or soluble fiber will cause the reduction of digestibility of the food or feed. Right? And some other, the heating can cause the reduction of protein digestibility. When you heat the protein, especially when there's a reducing sugar or like sugars are present or together, the amino group on the R group of lysine, that amino group will be called epsilon nitrogen or epsilon uh, amino group. Okay, they will be combined. They'll be making a bond with aldehyde, um, which is the glucose. Okay? Aldose, actually. The glucose is one of the aldose, and it will make a connection. And so this amino group on the R group of lysine will be attached to glucose, but then the trypsin cannot find this. Okay? So the trypsin cannot do its job. Okay, so this heating causing the combining of glucose to lysine in the middle protein, which is called Maillard reaction. It's called together as a Maillard reaction. When Maillard reaction happens, the protein digestibility will be reduced. And the lysine actually also cannot be utilized to make a protein either. Right? So Maillard reaction, the benefit, there's benefit. It is called caramelization. It will give a very good flavor, but in terms of protein utilization, it will cause the negative impacts. Right? And some other cases, so heating or again heating, and also alkaline treatments will cause the changes of L amino acid to D amino acids and causing the racemation. And if that happens, racemation of the amino acid in the middle polypeptide or protein, and then the protease cannot recognize that and cannot react on it. So that's another reason for reducing the decreasing protein digestibility. Okay, now we talked about what happens in the small intestine. Now we can move to the large intestine. In general, what happens in the large intestine, all the undigested matter from the small intestine will be moved to the large intestine, and that's where the water will be absorbed, removed, recycled, and also some minerals can be absorbed through the large intestine, and some small volatile compound like acetic acid or propionic acid can be absorbed through the large intestine as well. This is where the microbial fermentation happen. Um, so there are a lot of stuffs are generated from this fermentation, including carbon dioxide, methane, hydrogen, volatile fatty acid or short chain fatty acids, ammonia, hydrogen sulfide, and so on. Some example in relation to protein fermentation. Okay, so if some part of protein is undigested and moved to the large intestine, um, microorganisms, bacteria, can utilize lysine, and this can be quickly converted to some other compound or called cadaverine. Cadaverine is causing a very, very strong, unpleasant odor, the odor of the dead body, and uh, caused that is caused by the conversion of lysine to cadaverine by microbial fermentation. Another other amino acids like ornithine and arginine can be fermented by and utilized by bacteria and be converted to putrescine. Putrescine is causing also the unpleasant smell, odor. Right? But interestingly, actually, the cadaverine and putrescine, even though they have very bad smell, they have a very good industry application. Both of them are used for making bioplastic, okay? As an example. Tryptophan can be utilized by bacteria, be converted to scattles and indole. They also cause the bad odor. And cysteine also be converted to mercaptan will be the cause of bad odor as well. So the compound, when the undigested matter is fermented in the large intestine, and they often have bad odors because of the production of a cadaverine, putrescine, scatol, indole, and mercaptans. Right? Mercaptan contains sulfur. Right? Also, some other things happens in the large intestine by bacterial fermentation. Histidine will becomes histamine, and tyrosine will be converted tyramine, and tryptophan converted something called tryptamine, and those compounds can be absorbed into through the large intestine and cause body reactions. Okay? So large intestine has some important role here too. Okay? We should not ignore talking about ammonia here. In the large intestine, that's the source of ammonia because uh, urea is made from the liver. It's supposed to go to the kidney and excreted as a part of urine, but some part of urea through the blood circulation will be transferred to the large intestine and provided into the lumen of the large intestine. That's where the bacteria will utilize urea to convert it to ammonia because bacteria has a very effective enzyme called urease. It will hydrolyze urea into ammonia. And then this ammonia will be used by bacteria as a source of nitrogen to make the other amino acids and so on. And or the ammonia can be absorbed again, going through the blood circulation and used for the production of urea in the urea cycle. So this ammonia and urea is cycling going around in the body as well. And large intestine has some role here. Okay, so this is all I have in relation to protein digestion. So the second part, we talked about what happens in the small intestine and the large intestine. We spent good time talking about the enzymes as well. Okay, right, thank you. Well, before I finish, I'll stop sharing the screen. I'll just say bye again to you. Okay, I'll see you soon again.